Hello everyone, and thank you for joining this webinar on patient-derived organoids for drug development and screening, presented by Dr. Sylvia Boj, Scientific Director at the Hub. Dr. Boj earned her PhD under Dr. Jorge Ferrer in 2006, investigating the functions of HNF1 alpha and 4 alpha in pancreatic beta cells. She moved on to do a postdoc in the lab of Hans Clevers at the Foundation Hubrecht Organoid Technology in Utrecht, the Netherlands, where she established human pancreatic organoids from tumor resections. Dr. Boj joined the Hub as a group leader in 2014, leading the cystic fibrosis and pancreatic cancer programs. In 2016, she was appointed Scientific Director of the Hub with the ultimate goal of transferring the scientific advances of organoid technology into the realms of drug development and precision medicine. A warm welcome to you, Dr. Boj, and thank you very much for presenting your work for us today.
Thank you. So first, indeed, I would like to thank to Jean-Marie for this nice introduction, and also to STEMCELL Technologies for giving me the opportunity to present our work to all of you today. And I would like to start also with introducing our organization. So Hubrecht Organoid Technology is a non-for-profit company founded on the pioneering work of Hans Clevers in Utrecht, the Netherlands, who discovered methods to grow stem cell-derived human epithelial organoids. And our main goal is to translate organoid technology from the bench to the bedside. And at HUB, we think that indeed organoid technology can revolutionize the drug development and patient treatment. And why? Is because for the last decades, when a drug has been developed, the preclinical development stage was mainly using the models available until now that were conventional 2D cell lines or animal models. And then once the drugs passed this first phase, had to go into a clinical phase to prove their efficiency and efficacy in human patients. And it's quite striking to realize that a very low percentage of drugs have reached the point that can be used for treatment of patients. So what we think that organoids can do is to reverse this and bring almost each patient all the variety in genotype and phenotypes from the patient population into the lab, into the bench in a form of organoid. So in my talk, I will talk about the adult stem cell-derived organoids. I will have a small introduction about scientific key discoveries in the laboratorium of Hans Lab that allow or make possible the generation of the organoid technology. There are also some practical aspects of how to culturing human intestinal organoids, especially for CF experiments, and also some examples of how we can model, in this case, cystic fibrosis disease using organoids. So for the last 2 decades, the lab of Hans Clevers has been focused on studying the intestinal epithelium for several reasons. One of them is that it's tissue which Wnt signaling has a very important role in maintenance of homeostasis, but also because it's a very interesting tissue to study adult stem cells, since the intestinal epithelium in any mammalian needs to be regenerated every 4 to 5 days. So there is a formation of new cells constantly. It's also a very simple tissue in terms of architecture. So there are 2 main domains, the crypt domain, where there is a high proliferation rate of cells, and then the differentiated domain, the villi, where the differentiated cells are present. And in this model, there is a very hierarchical structure, very well defined, where stem cells give rise proliferative progenitors that, depending on the signaling that they receive, they can generate the different cell types present on the intestinal epithelium. And in the early 2000s, at that moment, it was assumed that the stem cells were located in this +4 position based on some labelly retaining experiments. So Hans Lab has been focused on Wnt signaling for a long time, and I'm just summarizing a lot of years of experiments in this slide, but among different genes which expression was highly dependent on activation of the Wnt signaling, LGR5 appear as one of highest Wnt-dependent genes, and Nick Barker, former postdoc of Hans Lab, get interested on this gene, and he observed that when staining for this, for the expression of LGR5 protein, he could see that these very tiny small cells in the base of the crypt were labeled, so there was expression on this area of in the crypt. And then these cells, as the staining for Ki67 shows, were cycling cells. And indeed, this image was quite striking when going back to some old data in the 70s. Professor Leblon already identified these crypt-based columnar cells located between the panet cells, and already in the 70s he postulated the possibility that these will be the stem cells of the intestinal epithelium. So Nick generated mouse model to try to identify and be able to isolate the LGR5 expressing cells. So in the locus of LGR5, he generated a transgenic knock-in where the expression of GFP, the fluorescent protein, but also the inducible CRE were expressed. And when the animals were generated and the intestine of these mouse were isolated, he could nicely observe how the pattern expression of the GFP under the regulation of the LGR5 locus was expressed on these columnar cells in the base of the crypt. And again, this resembles exactly as the drawing of Leblon about the crypt-based columnar cells. So he was able to identify the LGR5 cells in the crypt, and with the idea to demonstrate if indeed these LGR5 positive cells were the stem cells of the intestine, he took advantage of the model generated since under the locus of LGR5, the CRE-ER protein was also expressed. This CRE protein is able to recognize LOXP sites and then recombine DNA. So if he generated a mouse crossing this LGR5 with this LAC-Z model in a way that in the presence of tamoxifen, of course, because this CRE is an inducible CRE, the cells that express LGR5 will have expression of the CRE. This CRE is able to recombine this locus and activation of LAC-Z that can be measured by measuring the activity of beta-galactosidase. So the progeny of these two mouse will generate LGR5 positive cells with this blue marker because of the expression of beta-galactosidase. But the most interesting thing is that this is a genetic marker. So once the stem cell has activated the expression of LAC-Z of beta-galactosidase, this change can be passed to the daughter cells. So with the idea that if these LGR5 cells are the real stem cells of the intestinal epithelium, not only the LGR5 cells, but any cell derived from this will also have the blue marker. So this is what the experiment that Nick performed, where he just gave a single induction of tamoxifen just to activate randomly a specific LGR5 positive cells in the intestine of the mouse. And then, as you can see, after 1 day of induction, he could already observe the presence of the blue marker on the base of the crypt, suggesting that few LGR5 cells were heated. And then 1 week after injection, he collected new samples, and then he could observe already that the presence of the blue marker not only on the crypt, but also among the villi of the intestine, suggesting that now this LGR5 cell was giving rise to all the differentiated cells present on the intestinal epithelium. And this mark was maintained after 3 and 6 months, suggesting that this LGR5 positive cell is the stem cell of the intestinal epithelium and was actively proliferating during all this time. Another hallmark of stem cells, of course, is that they give rise to all the differentiated cell types, and by these co-stainings of the X-gal positive stain with cell-specific markers like lobed cells, panet cells, and enteroendocrine cells, he could prove that indeed LGR5 was the stem cell of the intestinal epithelium. And of course, it's an adult stem cell because all these experiments are done with adult animals. So the fact that in the lab it was available an animal model where the stem cells could be isolated, it facilitates the isolation of these cells and, of course, the performance of all the possible characterization of these cells to understand what type of signaling receive these cells, what are the pathways that need to be activated in order to maintain the stem cell mass characteristics. And this provides a lot of information to the lab that Toshiro Sato took advantage of in order to identify what will be the signaling necessary to grow these stem cells in an in vitro system. And this was something that the lab was trying to develop with a lot of interest, because so far there were not available that many in vitro models to study to grow intestinal cells. So having the right cells, the LGR5 positive cells, and all the information to identify what signaling these stem cells should receive to maintain their stem cellness, he was able to put them in culture, give them the right growth factors, and observe how these cells were able to grow, and over time generate a structure that we define as organoid, where all the different cell types are present in the same way that they are on the intestinal epithelium in the animals. So from this single LGR5 stem cell, an organoid was generated, and he could identify the presence of all the different differentiated cell types. Again, being another proof that indeed this LGR5 was a stem cell of the intestinal epithelium. This is an image of a culture of a mouse small intestine organoids. And I mean, the most striking thing about this model is that Toshiro Sato was able to grow healthy, non-transformed cells and expand them in vitro without adding any factor to induce the proliferation. They can be generated from single cells, but of course the efficiency increases if entire cribs are put in culture in order to establish the organoid lines. A very strong collaboration of Hans lab with the lab of Edwin Kuppen has proven for the last years with several models that these cultures are genetically stable. Because of a problem of time, I cannot show all this data, but for intestinal and liver models, organoids that have been expanded over a year every week, DNA has been collected at day 1 and after a year, and the DNA has been sequenced, and it's been observed that these cells in culture don't accumulate more mutations than the ones that our cells can accumulate on our body. And something also very interesting is that, except for a couple of details that we will talk about them on the coming slides, these organoids can be treated as any in vitro culture cell line. So they can be expand, you can isolate easily DNA, RNA, you can do histology, you can cryopreserve, store your samples, thaw them, and keep expanding them for future experiments. So they became a very easy tool to develop all the in vitro experiments, but also providing a higher level of complexity compared with the conventional early lines. With, of course, the stressing point that there is not any other way to culture, again, healthy, wild-type, non-cancer, non-transformed tumor cells. So when Toshi starts the development of the organoids, of course, he starts using a mouse first because of the existence of these LGR5 positive mouse that allow him to take the right cells to find the right conditions to expand the stem cells. And in the case of the mouse, of the intestinal organoids from mouse, the only thing that he needed was to activate the AGF pathway. Since these are epithelial cells, it's important, these cells need AGF to grow. It was also important to block BMP signaling and then activate Wnt signaling at that time using just addition of a responding. And then there was also another key factor for being able to develop the organoid model is to the use of matrigels. So these are epithelial cells, so it was essential to embed them on this extracellular matrix that will allow the cells to maintain their polarized features. He found that quite soon is that in order to generate the same model for humans, and this is a representative image of an intestinal human culture, is that for culturing the human model, in the case of intestine, it was necessary to activate stronger the Wnt signaling. So a difference of the mouse small intestine, the human ones, apart from a responding, also need an external source of Wnt, otherwise they cannot be expanded. And it's necessary also to block not only BMP but also TGF-beta signaling, otherwise the cultures then have tendency to differentiate, that they don't proliferate. And indeed, he needs to add 2 more compounds, nicotinamide and p38 inhibitor, to maintain the stem cell mass of the expansion of the organoids. So I'm talking today only about intestinal organoids, but what we've seen in Hans lab is that every time that a model has been developed either for liver or pancreas or other tissues, normally the mouse models, when using mouse cells, we need less molecules to maintain the stem cell nest or the proliferative activity of the culture, as well for human. We need to activate much more, otherwise they have tendency to differentiate quite easily. So, of course, in Hans lab and Toshiro Sato developed the models for intestinal organoids, mouse and human, and immediately the lab Hans get focused on developing the same organoid model using other different tissues. So we have right now available models, organoid models for pancreas, and very soon they will be available for lung and breast, and there is people developing models for other tissues like ovarian or kidney. And the interest of the hub is not for mouse models, but for human, to try to develop biobanks so that we can grow organoids from different models. And since these models come mainly from patients, that we can link the clinical data to the organoid data. So this becomes also quite valuable model for studies and analysis. And then, despite that the first models were developed using healthy cells, immediately we saw the possibility and the interest to develop the organoid models for disease models like cancer or cystic fibrosis. So right now, in our organization, we are making a big effort generating these biobanks with organoids, and our main activities now are developing the models for cancer and cystic fibrosis. And what I will explain you now on the rest of the presentation is about our work with cystic fibrosis organoids. So for those people that is not familiar with this model, cystic fibrosis is a genetic disease that it's affecting 70,000 patients worldwide. The mutations have take place in the CFTR gene that give rise to a transporter. And something quite striking is that there are more than 2,000 different mutations that can cause CFTR. So I will play now a small movie to try to explain disease. CFTR, the gene mutated on the cystic fibrosis patients, is expressed in many tissues, in all the intestinal tract, stomach, intestine, large and small intestine, pancreas, liver. But the most common phenotype, or the one that affects the span life of the patients, is the lung phenotype. So what happens in these patients in a normal situation, because they have a functional CFTR, they have a fluid mucus that allows the cleaning of bacteria and possible infections. However, in patients with cystic fibrosis, this mucus is thicker due to the not activity of CFTR, and then tend to have more infections and diseases. So there are now available some compounds that I will talk about them later, that can be provided to the patients, and then these compounds are able to improve the activity of the CFTR transporter, either by bringing more to the membrane or to helping to open this transporter, and then this will translate it in recovering the fluid, more liquid mucus, and avoiding or removing decreasing the chances of infections and disease. So as I mentioned before, we are making a big effort to generate a collection of organoid models from cystic fibrosis patients. And right now what we have in our biobank is 219 different organoid cultures that include 92 of these 2,000 possible mutations related to CFTR. And this is just a small list of the most common mutations that we have represented on our biobank that are probably related. So the most common mutation is patients with homozygous mutation for A5FL. But then the other list of mutations that we have a higher copy has to do with the fact that we are located in Holland, meaning that it's served that in different regions of the world there is enrichment for certain specific mutations. And we have this biobank available for performing some drug development and clinical trial validation that I will talk later. So what I will do now is to present a couple of slides talking a bit more technically about how to culture these organoids and how to work with them, and to identify certain aspects to know if we have a healthy or happy organoid culture, because I assume that being hosted by STEMCELL Technologies, this webinar will be interesting for the audience. So since these are intestinal organoids, our sources are normally biopsies, where we just by mechanical dissociation, we can isolate the crypts. This is a representative image of an intestinal crypt that we embed on this matrigel and then put them in culture. And this is just an image of 5 days difference between an isolation and how these structures grow and generate new organoids. So definitely for the efficiency of establishing organoids from human biopsies, the most key factor is the quality of the biopsy, meaning that it's essential that the biopsy is deep enough that crypts can be isolated. Otherwise, if we receive a piece of tissue enriched with villi but not crypts, then it will be almost impossible to establish the culture, the organoid line. So indeed, this is how a healthy colorectal organoid looks from a healthy donor. So the organoids tend to be cystic and with very thin layer and cells proliferating. But indeed, because of the CFTR mutation, this is macroscopic phenotype of the CF organoids. And this phenotype is indeed caused because of the dysfunction of the CFTR receptor. So we know that indeed increase on cycle IMP it activate CFTR, and then there is this transient of fluid in the organoid, and this is what it has this balloon phenotype. And because of the absence of activity, these organoids cannot make these balloons, and then they have this full activity. And this is just a culture that has been passaged, and we normally passage these organoids by mechanical disruption, because as you can see, this is a representative image of how an CF culture will be ready to be disrupted for the next passage. So they reach such a high, big structures that just by pipetting up and down on a pipette, we just disrupt these buddings that the organoids show, and these little buddings are what we place on the next passage. And it's also quite striking to see how in 8 days from these little structures we can generate such a big organoids, suggesting that there is a very high proliferative rate on these cultures. Also, something very important that we've been improving for the last years is how to cryopreserve and store these organoids. So we've seen that we get quite high efficiency on recovering the organoid from frozen vials when we thaw them in a very high concentration density, very crowded. We let them be for 2 or 3 days to grow, to have a bigger size. But after we can observe that the organoids are growing, they're becoming bigger, they are proliferating, then we just dilute them to have around 20 to 25 structures in a drop of 10 microliters of matrigel to give them a space to grow and to expand as these budding structures. And something that we always have observed is that while the culture, when it's running, we can expand every 7-10 days. Normally, the first time that a culture is generated from a thawing vial, it takes between 15 and 17 days to have the right size. And that's something also very practical that we've seen that it helps for the cultures, is that we normally have these ratios between the volume of matrigel depending on the site of the well. So normally in a 24 well plate, we will place 50 microliters of matrigel. What we see is that dropping or generating little drops from these 50 microliters helps to the growth and to the happiness of the organoids, mainly because there is more surface available for organoids to be on the medium. So what we observe is that if we have very big drops or very crowded drops, too many organoids, they just cannot bud properly, or they even attach and die, because especially in the center of the drops, there is not that much growth factors and molecules to signal these organoids, and it's not a problem that they cannot go through the matrix cell, but just that the organoids in the edge are just uptaking most of these growth factors. So density, having the right number of organoids on the drop, and providing them to be as much available to the medium, increases the success of the growth and the quality of the organoids. So apart from generating these biobank, the utility of the organoids in case of cystic fibrosis is very relevant, because indeed the laboratorium of Jeffrey Beckman developed an assay using intestinal organoids to measure restorage of CFTR activity or modulation, and this becomes very relevant for patients because currently there are two drugs available in the market called Lomacaftor and Ivacaftor. That one of them is a corrector, so meaning that it's a small molecule that can help the non-properly folded CFTR mutated transporters to move to the membrane. And then Ivacaftor, also known as a potentiator, that can help to open the transporter present on the membrane for the transport of the ions. And these two compounds are the ones available now in the market for treatment of patients with cystic fibrosis, but it's important to realize that currently there are several clinical trials for new compounds from other companies. So I think that if 20 years ago someone would have told us that a disease like cystic fibrosis could be a druggable disease, we will be quite surprised. But now organoids can help to identify what patients should receive what drug, or if a drug indeed will have activity on improving the function of the CFTR transporter. So as I mentioned, the organoids were developed and happening in the lab of Hans Clevers at Hubrecht Institute, and then Jeffrey Beckman and his team in the Wilhelmina Children's Hospital in Utrecht was aware about these organoids, the possibility to generate them even from biopsies. And then he had this vision of thinking how to develop an assay using organoids to measure CFTR activity. And with this, he together with Florine Dekkers, they developed what we call the Forskolling-induced swelling assay, of this assay that consists just in breaking these organoids in these very small strips, and then put them in culture in a plate, incubated with drugs to test. So normally, if we are testing a corrector, since it needs to help the proper folding of the transporter to go into the membrane, we incubate it for 24 hours. If we are using a potentiator, we normally add it at the moment of measuring the assay. But after 24 hours, the organoids being incubated with the compounds, they are labeled in green with the calcium green. And then after receiving a stimulus, in this case is the addition of Forskolling that increase the cycle AMP. This activates the kinase that needs to phosphorylate the CFTR transporter, and then if there is a restorage of the activity, the organoids swell and show this organoid growth that can be measured by imaging in a confocal. So this is a representative image of one well of a 96 well plate with about 50, 60 of these little structures from a patient, homozygous del-del, where it's not been incubated with any drug. At the moment of start the movie, Forskolling is added. And then, as you can see over time, so this is a movie of, these are 1 hour summarizing 7 images. Nothing happened to the organoids. However, when the same organoids were exposed to 809 for 24 hours, and then 770 was added at the same time then Forskolling, we can see how the organoids swell. And this can only happen because either CFTR activity has been restored by the presence of these two compounds. So what you've seen now is just a movie generated by pictures that have been taken every 10 minutes over an hour experiment. And then what we do in order to analyze it, to translate this movie into numbers, is to measure this is a representative image of one well time 0, where we have recognized each object and what is the area of the organoids, and then the size that they reach, or the area that they have the organoids when after an hour. So we just have a program that can recognize these objects, measure the area, and then measure this, establish this initial area as a relative number of 100, and then measure the increase of this area over time for the last 16 minutes. And this is how normally organoid swelling on a FISH assay is represented. What you can see here is organoids from an A5 homozygous patient, where the condition DMSO is showing in white. So there was not, in this case, there was not any swelling after adding forskolling at the 5 micromolar concentration. Then when the corrector 809 or the potentiator 770 alone were added, it was possible to observe some swelling, but we can see that there is a bigger effect when the two compounds are combined in terms of organoid swelling. And another way to represent this data is just the area under the curve that we have in this measurement. And here we can see also clearly the impact of already adding a bit of potentiator or corrector on the CFTR activity, and how the organoid swelling is increasing. We can see a more than an additive effect of these compounds, since indeed the dotted line is showing what will be the organoid swelling or the area under the curve if we will sum the effect of the single compounds. So with this assay, we are able to measure the activity of any compound. And just to show you the reproducibility and also the robustness of the assay, here I'm showing you the same experiment. So this is an organoid line from an A5 homozygous patient, which assay was performed in 3 separate weeks. So the organoids were passed every week, and that week the assay was done. So if we look at the reproducibility of the replicates for the organoids are very, very good. And then the same organoid line has been passed over more than a year weekly. And then we perform assays at 6 months after. So these results are almost around 3 months after the organoid was established from a fresh biopsy. Then the vial was frozen, was expanded, and then cultured for 6 months and for a year. And you can see that the results and how the drugs are behaving on this organoid are maintained over time, meaning that we have a quite robust assay. And very important in terms of thinking on patients is that we are able to generate an organoid culture from a patient, maybe grow it now and expose the organoid to the available drugs, but the sample can be frozen, and if in 1, 2, 3, 5 years a new drug is in the market, or there is a development of new drugs, we can follow this organoid line and expand it, and then measure the activity of whatever compound we want to test, having the security that we are maintaining and representing the same activity that was observed when the organoid was coming from a fresh, isolated sample. We are now quite busy to try to, as I mentioned in the beginning, we are trying to translate all this into drug development and also patients. So in terms of CF, we have several activities running related to CF. So as I mentioned before, we are generating an organoid biobank that of course is available for both academic and commercial partners in case that experiments or tests want to be done. We also perform drug screen in-house for new compounds that companies are interested in assess. But we are also putting a lot of energy to see how this technology can be translated into the benefit of the patients. So what we are currently running together in collaboration with UMCU and many other hospitals here in Holland are retrospective and prospective clinical trials to validate organoids as a patient response. What we are doing right now is collecting organoids from patients, generating the organoids, but also exposing these patients to the available drugs to collect, to identify the clinical impact of being treated with the drug and correlate with the organoid data. to see if via one-to-one comparison we can demonstrate the predictive value of organoids. But already Jeffrey Beckman and his team have published some data that looks promising, and it's really was one of the reasons to really move us to perform these one-to-one clinical trials, where, of course, when they developed the FISH assay, they, in this case, they were exposing different genotypes, as you can see, to the available drugs. So we have the response for either treatment with only the corrector 809 or the potentiator 770, or the combination of both of them with F-del class 1 or F-del homozygous or gate mutations. And they could observe that, of course, there are differences responses, and, for example, the gating mutations can respond already to the presence of potentiator without the need of correct. So, of course, without being the same patients, but when looking, there is already some clinical data published showing the response of certain genotypes to some of these available drugs. So just to show it here, what they did was to compare what was the organoid swelling for all these different genotypes, and then compare to, so they could define like an area where, well, there is not that much swelling organoid, there is not that much going on. There is another area that it's very clear that the drugs have an impact on the organoid swelling, so they are restoring CFTR activity. And of course, there is also an area that it's a bit difficult to predict what will be the outcome. But then when they compare these groups, so the group that is having the higher clinical benefit by measuring on the increase of FEV1 compared with organoid swelling, they have almost a perfect correlation, suggesting that at least in terms of genotype groups, the organoids can predict or can correlate their response to swelling to the benefit of the clinical validation. So what we are doing now is indeed the comparison of one-to-one, because this is just data from patients in different locations, and these are organoids from a group of patients with the same mutation. And of course, something very exciting is that indeed here in Holland we have the first case of a patient that has been treated with CF drugs because of organoids. And this is the case of a young patient with a very rare mutation, because indeed there are only 2 patients in the world with this S1249R mutation, that the group of Jeffrey Beckman and Dr. Kors van End were able to obtain biopsies from this patient. And then, of course, as a CF patient, when it was exposed to forskolling, there was not any swelling, but when the organoids of this patient were combined with the 809 and 770 compounds, there was organoid swelling. And this was indeed the data that was used to convince the medical authorities in the hospital to treat these patients. So we have the first patient treated with organoids, and if you want to know more, indeed there was a report published on STEMCELL on 2016. What we are also doing is indeed, because organoids start running the first clinical trials based on organoids. And as I mentioned at the beginning, there are more than 2,000 mutations. 50% of the patients are homozygous del-del, but then the other 50% of patients are distributed around almost the rest of mutations, meaning that we have a lot of small groups of patients with the same mutation. They are not possible to run clinical trials with them. So again, the work of Jeff Beckman showed that a mutation that is very frequent on this area of Europe, the organoids of these patients were responding to the treatment with Orkambi, with 770 and 809. This is the results of this genotype in the area under the curve, and it is on the green area. And because of this, we are indeed currently running so in this case, the company Vertex ran clinical trial with a group of these patients in combination with organoid data to see if they will respond to their treatment and present this data to the appropriate authorities for the approval of this drug to this genotype. And also, and this is the last example I will show you, is that I've shown you a couple of cases where organoids were predicted positive in terms that we see organoid swelling and the patients respond. And we also had an example where organoids predicted the lack of response for the genotype. So this is a publication coming from Vertex Lab that was using conventional 2D cell lines where they generated CFTR molecules, CFTR genes carrying specific mutations that were present in different patients to see the response to, in this case, is ivacaftor, because all these mutations were considered gating mutations. And after seeing all these results, Vertex decided to run a clinical trial in order to include all these different mutations for the treatment of Kaleido, this 770. But on the genotype G97OR, there is an asterisk, because indeed in this clinical trial, these patients did not benefit for the treatment of Kaleido, despite that their experiments in 2D cell lines will suggested that indeed they should respond. So we received biopsies from these patients, from patients with G97OR mutations, in order to establish organoid cultures and run the FIS assay. And what we did was we collect from our biobank genotypes with a positive response on the clinical trial as a positive control. And as you can see here in this graph, this is the area under the curve when organoids were stimulated with 5 micromolar for scoline and 3 micromolar of ivacaftor. We can see how there is an increase in organoid swelling when they are exposed to the drug. Then we use as a negative control 3 organoid lines that were FFL null mutation. So it was expected that the presence of ivacaftor won't have that much impact on organoid swelling. And then when we performed the organoid swelling on the G97OR patients, we also observed that there was not organoid swelling. So the data coming from organoids reflected what happened in the patients. And indeed, despite that this mutation was initially identified as a gating mutation, indeed needs to be classified in a different way, because as you can see on this Western blot, when we isolate protein to do CFTR Western on these patients, we could not identify the CFTR band. So this is another example where organoids can have provided the same response that has been observed in patients. So we are making a big effort to work not only with Vertex, but with any company developing drugs for CFTR patients, because as I mentioned, we have a lot of patients with very specific mutations that thanks to organoids can benefit in terms of having access to drug or a compound that they won't have access through the normal classical clinical trials methods. And with this, I would like to finish my presentation, first giving the thanks to our founders, Canave, UMCU, and Hubrecht Institute, also the funding that we've received from the insurance companies in order to run all the trials and validations we are doing with cystic fibrosis organoids. Of course, the group of Hans Clevers, especially Nick Parker, Toshi, that I presented their work, but also the amazing team of outstanding PhD postdocs and senior scientists that Hans has in his lab, of course, also for Hans, and for the entire Hub team, but a special mention, of course, to the CF team working very hard in order to perform all these validations, and of course, a special mention to our very close collaborators at the Children's Hospital, UMC, Jeffrey Beckman, his team, and also Korsman and also his team. And thank you very much to all of you for listening to me.
Thank you, Dr. Boj. That was very inspiring. I was particularly struck by how the proscolin assay is used for the drug screening and the intersection of the prediction of drugs and then the fast validation that they provide. And also, I know you're working with the health insurance companies in Holland for screening all CF patients. Do you think that will be a one-off, or do you think once you've more or less cured all CF patients in Holland, you'll move on to other diseases?
Sure. I only had time to talk about CF, but we are also making an effort right now for cancer models. So we are also starting to use organoids to see if they can predict patient response for colorectal and pancreatic cancer. And of course, because of the potential of CFTR being a monogenetic disease and a bit easier model than cancer, that it's much more complex, we are also looking for other monogenetic diseases that are expressed on the tissues where we can develop organoid models. And right now I'm thinking on any metabolic disease from liver, for example.
Okay, so it has more or less endless uses, I guess.
We are trying.
Yeah.
I mean, we need to run all these validation trials and see that they work, but at least the next years we're going to make a big effort to make sure that we can identify to what disease or to what models organoids can contribute for the treatment of patients.
Thank you. How can I order organoids from the bank, and are they shipped frozen or in live culture? So could you speak a little bit about how you collaborate with the researchers?
Yeah, so we are now working in making available a kind of catalog where people can see all the different biobanks that we've generated, so not only CF, but we also have cancer and other diseases. I mean, right now the easiest and what we are, they should be able to contact us through our website, Hub for Organoid Technology, and then the organoids are available. The only thing is it has a certain cost, and it's different if it's an academic or a company collaboration, but we are a not-for-profit organization, so we need to cover at least expenses of generating the organoid lines. And something that it's still required now, despite that we are trying to reduce the time, is that since these organoids are collected from patients with a specific patient consent, we need to get the approval of the ethical committee from the local hospital in order to distribute the samples. So it's not a problem that the samples won't be provided. We've not get any cancellation of a request, but it extends a bit the time that the organoids are available because we need to go through these ethical procedures.
Okay, thank you. So that's good for everyone to know. Another question is, how do you ensure the consistency of quality between different lots of organoids if you have them in a bank?
I mean, of course, we see certain difference between different samples. I mean, what we can clearly notice is that at the beginning, our biobank was generated from young patients, so patients that were with cystic fibrosis under 12. And then lately, we get consent and permission to start collecting samples from adult patients. And we can already see that, the younger the patient is, the organoids grow much faster and much easier than if the patient is very old. Of course, over these 200 organoid cultures, maybe 0.01% are difficulties to grow, and they tend to differentiate despite that we try to culture them. So yeah, since they come from different patients, we are able to observe a certain variability or capacity to expand or to grow. But it's definitely the samples we have available in the biobank are the ones that we are sure that they can be distributed and people can expand and do experiments with them.
Thank you. So one question here is touching upon the Forskollen swelling assay. Have you developed other assays, and which are they, to quantitatively measure the phenotypes in the organoids, and maybe not just for cystic fibrosis, but also for cancers?
Right now, our main activity is to translate organoids to drug development and clinical trials, so we are not that much involved on the new development of assays, at least for CF, and because we have a very close collaboration with a group of Jeffrey Beckman. Indeed, he is working on this, on trying to define other ways, other methods, other type of assays to study the interaction of drugs. So what we do, it's more like when an assay is established, we just take it and then develop it to then perform screens, larger scale. And for example, for cancer models, the assay that we have is just the cell viability assay. So we can perform cell viability assays with cancer organoids, and what we've put an effort has been more maybe on the scaling up, the expansion of organoids to be able to do middle throughput screens, be able to screen 10,000 compounds using organoids, but not in the assay itself. And of course, we have now a very dedicated team developing models for toxicology, for example. But these are things that we are right now developing, and there is nothing in place yet. But of course, as soon as it is, we will be very happy to share it with the people.
Thank you. So I think we'll have time for one more question, and that is, are the media conditions the same for both the human wild type and the cystic fibrosis organoids? And if so, why do they look so different? One is budding and one is not so much.
The difference is indeed the fact that in the cystic fibrosis patients, the CFTR is not active. So the composition of the medium that we use to grow the organoids increase cycle IMP. So we have, wind and other factors can increase the cellular cycle IMP. And this is the signal that the kinase, that phosphorylated CFTR, receives to open the channel. So CFTR is functional in the wild-type organoids, and then there is this interchange of ions and water, and it makes an osmotic pressure, and this is why the wild-type organoids are cystic. However, because on the patients with cystic fibrosis, this transporter does not work, the organoids cannot swell. There is not this pressure, and then they just grow, making these folders that remind more like the culture of the mouse intestinal organoids rather than the human. That is because of not the functionality of the CFTR receptor.
Okay, thank you very much. So this concludes our webinar. Thanks again for your interest, everyone, and enjoy the rest of your day or your evening.